that actually fixed it? I'll be. Okay. Last time I actually barely remember what we did last time, so probably like you guys. Anyways, oncogenes, these are going to be genes that can induce cancer under appropriate circumstances. An example of this is the mutated RAS genes. I think Burkett was mentioning you guys are actually doing RAS right now, aren't you? Or we're doing it just like a week or two ago? Anyways, there you go. Proto-oncogene genes, these are going to be genes that can be converted into oncogenes through mutation. So this would be, for example, the unmutated RAS genes. If you do a mutation on them, they can then be the mutated RAS genes, right? In which case, that'd be an oncogene. And again, under the correct conditions, appropriate circumstances, they can induce cancer. Again, even if you have the gene for cancer, that doesn't necessarily mean you are going to express cancer. Okay? Tumor suppressor genes, these are going to be genes that can contribute to the cancer development only when both copies of the gene are deleted or damaged. And so some examples of that are going to be genes that, some phosphatases that terminate the EGF signaling. Remember, the EGF signaling is going to be a pretty typical place where you could get cancer, right? Because it's growth factor. What do tumors have to do? Grow. Cancer has to grow. No growth, not a lot of problems. Yeah. Okay. By the way, does anybody know whether you should ever get a full body scan? Shouldn't we just full body scan everybody all the time? No, it's a really bad idea. If you full body scan, you'll have a dozen different little tumors, and they're meaningless. Okay? They just are not meaningful at all. And you'll come up with dozens of things, and the drugs that you give them are going to kill them, not the tumors. Okay? Anybody ever heard of the death spiral, by the way? No? Oh, shit, I really should tell you guys about this. Okay, let me break that one to you guys, okay? Sorry. So the death spiral is where PAs, doctors, they start prescribing drugs for something. For example, my mother was actually just put on a death spiral as of December, January. And I ended up stepping in and modulating her drugs and getting her changed on doctors and stuff. So what they do is they give you a drug like gabapentin, okay? The gabapentin then starts causing your blood pressure to drop every time you stand. And so they give you a medicine to raise your blood pressure. They give you a medicine to accompany that for a sodium channel uptake. They give you a medicine for your pain on top of that, which causes even worse effects. They go, oh, your pain must be causing this, and then they increase the gabapentin to double it. And all of a sudden, you're at the point where you can't even move from a fetal position, and your blood pressure is in the 200s. Okay? And guess what they try to do? They never take anything away. They just continuously put more drugs to try to take care of the side effects, which creates more side effects, which they give them more drugs. And eventually, it spirals out of control until you kill someone. Okay? And so if you ever have a situation where you're on you've within 1 year added over a half dozen drugs, you should really sit back and go, does this make sense? Or are you assigning drugs repeatedly to take care of things that could be side effects? In which case, you've put somebody in a death spiral. They will eventually die from it. Okay? Because all medicines have what? Side effects. All medicines are poisons. All poisons can be medicines. Okay? Okay, so you gotta be really careful about that. And I don't know what it is around here, but apparently that's really common. Because when we went to Vanderbilt, they're like, yeah, we see people all the time for this shit. And I'm like, oh God. Anyways, that was important. It's probably the most important thing I told you guys. I've done my job, we're done. Okay, so the monoclonal antibodies can be used to inhibit signal transduction pathways activated in tumors. Mutated or overexpressed receptor tyrosine kinases are frequently observed in tumors. Some of the examples are overexpression of the epidermal growth factor receptor, which would be the EGFR, in breast cancer. Remember, it's not just the EGF that could be overexpressed. You could be overexpressing the receptor as well. Okay? An overexpression of the receptor would also cause increased binding, right? And increased signaling. Okay? So a therapeutic approach is going to involve the monoclonal antibodies to the extracellular domains of receptors. Centucubamate, excuse me, let me get me. Okay. Centuximab treats a colorectal cancer by competing with the EGF for the EGF binding site on the EGFR. Trastuzumab treats breast cancer by blocking HER2 and another EGFR family receptor. By the way, normally what you would do with things like this is you can actually break them further apart if you would like. And you can go, it helps if you know where the root is, like what they're referring to. But you can actually break these further apart into trastuzumab if you need. And people will know what you're talking about, though it will not be as fancy, okay? If you ever have difficulties. Okay, so this is the cetuximab. It's going to treat the colorectal cancer by competing with your EGF for the EGF binding site on the EGFR. And you can actually see that over here. This is the trastuzumab. It's going to treat your breast cancer by blocking the HER or another EGFR family member. And right here you can actually see these figures of it fluorescing right there. Okay, so the protein kinase inhibitors can be effective anti-cancer drugs. By the way, a little interesting side note. I didn't realize that they used fluorescent spectroscopy in the spinal taps now. So that's interesting. They don't just use X-ray now. They use fluorescent spectroscopy as well. Huh. Learn something new every day, right? Okay, so the protein kinase inhibitors, these can be effective anti-cancer drugs. The majority of the chronic myelogenous leukemia, CML cases result from chromosomal translocation, which the tyrosine that tea really messed up my mouth. The chromosomal translocation, which is the tyrosine kinase encoding gene, which is CABL, on the chromosome 9 is going to be randomly inserted into the BCR gene on the chromosome 22. And this results in the overexpression of the BCR ABL fusion gene. And so right here is your chromosome. This is the CABL gene. We've translocated it. Now it's going to be next to the BCR gene. Everybody see that okay? Okay. And so that was the BCR right here. So we have translocated that. That's the translocation. Notice we've chopped off some of this yellow with it. And so this would be the BCR ABL gene. This one right here would just be the BCR gene. Okay? All right, so the drug amitinib inhibits the ABL kinase by blocking the ATP binding site. This right here is the amitinib, otherwise known as Gleevec. And right here, it's inhibiting that binding site right there on your ABL kinase. You can see that very nicely. So Gleevec is going to be the highly specific inhibitor of the BCR ABL kinase that's used to treat CML. It's going to occupy, again, that ATP binding site right there. It'll target tumor cells specifically, and this will help to minimize the side effects. Okay, so you guys do know that whenever you're trying to treat cancer or tumors, that basically every treatment that you can use on cancer or tumor will have some effect on the person, right? Because it is their cells that you're trying to kill, right? And so realistically, all tumor cancer treatments are treatments that you're trying to kill one thing faster than another, okay? You're trying to kill the tumor faster than you kill the patient, okay? If you have more specificity, you can have a little bit more leeway, okay? And the majority of this specificity is going to be targeting the fact that the tumors and cancer is typically going to be taking in a lot more stuff because it's trying to grow a lot faster, right? Okay, so sensory systems are going to be based on specialized signal transduction pathways. Most odorants are actually small organic compounds with sufficient volatility because they have to be able to get into the gas phase or lift it up into the gas phase to get to your nose, right? And so the shape of the molecule is going to determine your odor. This is going to enable interactions with specific receptors. There is functional groups that are also important, but a large portion of odor is the shape. There's a bit of it that's the functional group has to, of course, be hydrogen binding where you're supposed to be hydrogen binding and everything else, right? But it's the normal protein enzyme interactions that we're looking at. These right here are some different odors that you may recognize. Here we have ginger. This is rose. It's called geraniol, which is the rose. If you make geraniol, it smells like roses, of course. This right here is, does anybody recognize benzaldehyde, by the way? No? You've used it. Everybody that took my Organic Chemistry 1 used this. Yeah. And that's almond. That's why it smelled a bit like almond when you guys were using it. Yeah. This is skunk, by the way. Notice it's a thiol, but it's a fairly heavy thiol. This is why it tends to stick to you pretty well. Anyways, this is carvone. This is R-carvone. This is S-carvone. Notice the only difference here. You guys notice the difference? Right here. Yeah, forward versus backwards, right? Yeah, they're just mirror image enantiomers. And this is one of the kind of fun cases of it where, you get spearmint and carvone, depending on which side. There's also cases where they're poisonous. One side versus the other side is not poisonous. It's in fact like a food. Anyways, so the olfaction is going to depend on the CAMP, GTP, and a G protein alpha subunit called G, and then OLF for olfactor, right? Olfactory. This suggests the involvement of the 7-TM receptors, of course, because those look like all the parts of a 7-TM receptor, doesn't it? Except for the receptor. Okay. The human genome encodes approximately 400 odorant receptors. These are known as OR for the odorant and then receptor, right? Okay. And so each olfactory neuron is going to express only a single OR gene. And literally, we just have a single thing that will detect this sort of shape combination of hydrogen bonding, hydrophobic, hydrophilic interactions. Okay? This is why, by the way, if you ever smell thiol, skunk smells like what? What if you open a gas tap? Does that smell like skunk? Yeah. Rotten eggs? Smells a bit like skunk, right? This is why all the things, it's very difficult to tell the difference between some of these things, like rotten eggs versus the gas tap versus skunk versus rotting flesh versus etc., right? Because we only have 400 receptors. That obviously is not everything we're going to smell, right? Okay, so this right here is that olfactory signal transduction cascade. It's going to be similar to that for the beta-androgenic receptor. That was why we used the beta-androgenic receptor as like a case study, right? Showing the overall 7-TM receptors and how they work. Okay? And so what's going to happen is your odorant is going to bind to our OR. Remember, that's the odorant receptor. This on the neuronal surface, this is going to initiate a signal transduction cascade that's going to result in an action potential. And in this case, we're not so much triggering something inside of the cell, at least not as our final action. We're triggering an action potential. Does anybody remember action potentials? They're what sets off the next neuron, right? Okay. And so we're going to set off a neuron sequence that eventually will hopefully reach your brain, and your brain will go, skunk. And then you'll scream and run away, if you're smart. If you're not smart, you'll stay and get sprayed, okay? Okay. So again, you're going to have your rise of your CMP from this pathway. It's the normal pathway we've been talking about, right? Remember, this is the adenylate cyclase right here being acted via our G olfactory. The calcium and other cations are going to flow into this cell. This is going to depolarize the cell and initiate an action potential. And again, that's that action potential CAMP-gated ion channel right there. You starting to see how the pieces were coming together? Yeah. We had to do them in pieces, but now we got them. Okay. So this is actually the mouse nasal epithelium. This is loaded with Fura2 and treated with odorants one at a time. Neurons containing an activated odorant receptor were detected by a change of fluorescence due to this presence of the Fura2 here. The OR responsible was identified by generating cDNA from the mRNA that had been isolated from that neuron. And so right here we got some of the different compounds that were triggering those receptors. Okay? Does anybody know what this I right here means? Okay, so a lot of these, there's parts of it that don't really matter. Okay? So for example, you can have any number of CH2s in here, from 2 of them to 7 of them, and they'll all fit into that receptor. They'll trigger that OR. Okay? This part of the structure is the only part that matters. Okay. Everybody okay with that? Making sense? Okay, and so we have for example, carboxylic acids. These trigger something. That's why when you smell like acetic acid, formic acid, they all have that sort of stinging sense in your nose and smell a little bit like grassy or something. This is the alcohol. This is why methanol, ethanol, propanol all smelled kind of alike when you're in lab. It's the same receptor. There's not really much of a difference here, right? Anyways, these are some different ones. Does anybody have any questions about those? Okay. Note that this is mouse nasal epithelial. We don't get a chance to really work on these things with humans as much, because that would be unethical. At least that's what they tell me. All right. So different compounds are going to result in distinct patterns of olfactory receptor activation. Almost every odorant activates a number of receptors, not just one. This is why you have gradients, and you can train yourself to be able to tell the difference between different, like alcohols and stuff. And there are people and animals that can do so. Okay? But what happens is, unless you train this sense, and heavily so, like you won't be able to tell the difference between different nuts and stuff very easily. It'll be very difficult. Anyways, so you have a number of receptors. Each one of these can activate. Almost every receptor is going to activate it by more than one odorant. Each odorant is going to activate a unique combination of receptors. And here you see these different receptors, and these are the compounds that are activating them. And see the shading? That's how much it activates them. And how you identify these is then that combination of receptors would send signals to your brain. Your brain would then resolve those signal combinations. Okay? Which is why it needs training if a human wants to try to do that to any degree. And like a dog needs training, everything needs training. All the animals do. Because it's not just as simple as match. It's done. So color resistance is... It's triggering. Yeah. And so, for example, this carboxylic acid triggers this receptor very strongly. But the other ones also trigger just weaker. Mm-hmm. Yep. And that has mostly to do with binding. Okay, so this is going to be your vision. This is going to rely on a specialized 7-TM receptor to signal in response to absorb light. Your vision is going to be based on the absorption of your visible light by photoreceptor cells in your eyes. For women with hyperchromism, it's not just visible light. And for many women, they actually have little bits of UV that they can see. Damn superpower. For men, we're lucky if we can see that whole range. So, if you can see all of that, you're doing pretty good. Admittedly, it is a bad projector. Notice that visible light is over a very, very small range. Human eyes cannot work very well, actually. So, rods and cones. You all have heard of rods and cones before, right? Okay. And what type of receptor were we talking about? 7-TM. It's the 7-TM that's going to cause the cascade later on, okay? Okay, so vertebrates are going to have two kinds of photoreceptor cells. We have our cones. These are going to function in bright light. They're going to be responsible for color vision. We have our rods. These are going to function in dim light, and they do not perceive in color. An interesting thing is your rods, in the case of human eyes, are much more concentrated around the outer edges and can see your quick dynamic vision on the sides. Much better. So if you're ever looking in very dim light, look out the corner of your eye. Okay? Okay, so the rod cells, these are going to have elongated structures. These are going to include stacked discs where the light absorption is going to occur. And so these are the rods. You can see why they're called rods, because they look like rods. And literally they're rod shaped with stacked discs, right? That makes a lot of sense. Okay, and these are what the cells look like. Anybody tell me what this part over here is? Looks oddly round like a nucleus, doesn't it? What do you think it is? A nucleus. And don't forget, this isn't a nerve, so what part is this? That's going to be your synaptic junction, right? Yeah. Everybody okay with that? Fantastic. Okay, so the photoreceptor molecule in rods is actually rhodopsin. That's the actual molecule that does this. Rhodopsin is going to be the photoreceptor that absorbs light efficiently in the middle of your visible spectrum. This is actually why we have the visible spectrum, and this is why people's vision starts fading out towards red and towards violet, if you're ever curious. You guys ever looked at something truly violet? Or like really, really dark red? No? Okay, sometime get a chance to do that, and your eyes will start hurting. It's really a struggle. Okay? So this is going to consist of a protein opsin linked to 11-cis retinol. This is a prosthetic group via a lysine residue. So this right here is the 11-cis retinol right there. Okay? And this right here is the 11-cis retinol portion. This is then bound to the lysine right here. Notice, by the way, this is a shift base up here. Everybody remembers the shift base reactions we talked about last semester? Kinda? A little bit? Okay, it's okay. This is not super important. Okay, so this is a part of the 7-TM receptor family. The rhodopsin is going to absorb light effectively towards the blue end of the visible spectrum, and that's why it actually has this pattern for this extinction coefficient. You guys remember your spectroscopy portion of a visible light? You did in like Gen Chem mostly, but we got a little bit of it later ones. Okay, so the extinction coefficient being how well something absorbs that light, right? And so this is actually the center where you see best. By the way, that's why police lights are blue. It's because it's very easy for you to see. Okay, so the light absorption is going to induce a specific isomerization of the bound 11-cis retinol. Your light absorption is going to result in your isomerization of your 11-cis retinol group of rhodopsin to its all-trans form. And so right here we had the 11-cis form of the retinol. We absorb the light, and what do you notice? Well, that flips, doesn't it? See that flipping? Right there, along the red bond. Okay. Was cis on the same side, now it is trans. And so the all-trans retinol here is going to have a different distance between these groups right here. See that? It flips. See, it was over here. The shadow here is this cis one. And then the trans one is up here. And so we flip that group up 5 angstroms, right? Like a little switch. Like a little light switch. Okay. And so, again, that's that Lys residue. It's going to leading to the close of your ion channel and the generation of a nerve impulse. This right here is what it looks like. So we have the light coming in. We had rhodopsin bound to our 7-TM receptor. Our light is going to trigger the 7-TM receptor right here to modify that channel, right? Close that channel. Okay? It will eventually close that channel. So transducin is going to be the heterotrymic G protein that's going to activate a specific cGMP phosphodiesterase. This being the phosphodiesterase, by the way, down here. That's going to hydrolyze the GMP to cGMP, cyclic GMP to GMP. This is going to cause the cyclic GMP-gated ion channels to close, and that's what was being referred to here, the ion channel to close. That's here, that we're going to be closing this ion channel, thus propagating a signal over to the art. Okay, so this is not really something that I could go all the way into. It's just, it's its own science field. But you know you have a nerve center in the fore part of your brain that gets all these signals from your eyes and then assembles different like little shapes and stuff in different colors. Yeah. We don't actually see what we are looking at all. What comes in is a very chopped up version of it. Okay? If you want to talk about this sometime, we can do that during office hours, but I don't have time to go into that. Okay. So again, this is going to be activated by the all-trans form of the rhodopsin. This being the all-trans form after we've absorbed light. Okay. So what's happening is our light's coming in, rhodopsin is getting activated, right? It's swinging that a 5 angstrom distance. That triggers our 7-TM receptor right here. Okay? This is known as the transducin, right down here, which is the G protein is essentially, it's a G protein. They really missed the mark, didn't they? They could have done like G-Viz for vision or something. Yeah, but whatever. I didn't make that choice, I guess. So GDP is then going to be converted. So GTP going to GDP. This is going to give you, again, your same alpha unit going off with, again, GTP bound into it. That's from the transducin down here. This is then going to activate your phosphodiesterase. This is going to convert the cGMP to GMP. Now, the cGMP gate needs cGMP to activate. And so if we're reducing the cGMP, if we're getting rid of that, what's going to happen to our gate? We'll close it, all right? And then that's going to cause your propagation of signal. Anybody know why you'd want to close off rather than a pump through system? What can you do faster, stop or start? Stop typically, right? Yeah. It gives us a little bit of an edge, though we're still kind of slow for seeing stuff. case you're wondering, cats are actually like 4 times faster than us. But so cone photoreceptors. The cone photoreceptors are going to be members of the 7TM receptor family. This is going to use the 11-cis retinol as their chromophore. The signal through the pathways are going to be identical to that are used by the rhodopsin. Humans have 3 distinct photoreceptor proteins with different absorption maximas. And so these are the 3 different absorption maximas. Anybody realize what's going on here? How do you print things? What inks do you need? Blue, yellow, green? Or blue, yellow, red, excuse me. Okay, so blue plus yellow is green. Yeah. We're actually targeting our photoreceptors when we're doing that stuff, in case you're wondering. Okay. We don't quite target them because we want a little bit larger of a range, but overall we do. So again, that's why we see the visible lights as we do. Yeah, it's that combination of blue, yellow, red essentially, though we're actually doing a green color. It's not quite yellow. Okay. So the humans, there you have that 3 distinct photoreceptor proteins. These are going to have different absorption maximas. Each cone is going to express only one of the photoreceptors. Most wavelengths of light will activate more than one type of photoreceptor, and that's why we get the different shadings. It's how much they're activating each photoreceptor. Okay? Okay, and so the hearing, did everybody understand the vision stuff enough? Okay, I know we're kind of packing them in really quick, but yeah. So next is hearing. This is going to be the hearing. It's going to depend on your hair cells. These are going to use mechanosensitive ion channels to detect tiny motions, because that's what sound actually is, right? It's micro motions of the particles. So the cochlea is going to be a fluid-filled, coiled membranous sac. The hair cells are going to be specialized neurons inside the cochlea that detect sound waves. This is going to contain a hexagonally shaped bundle of about 20 to 300 hair-like projections. These are called stereocilia. Notice the name right there, they're cilia, but they're set up in a stereo, as in like stereochemistry, as in stereo, like stereo audio system, meaning the dimensions matter, right? 2 or more speakers so that the distance matters. Okay, so the mechanical deflection of your hair bundles is going to create a change in your membrane potential of your hair cell, and then we can sort of detect the propagation of those changes, and that's how we would then detect what sort of sound we're hearing. Okay? Think of it a bit like you can rub on a, anybody ever done one of those, they have like the thin hair projections and like you can make pictures by just rubbing on them, the felt drawing things? Everybody seen one of those? No? Okay, but that didn't really work. That's a great example of this. Right? It really works. Those old 1970s felt drawings, they were neon usually. Yeah, anyways. It was before my time too. All right, so yeah. Here's where we're looking at. Hearing is mediated by bundles of the stereocilia on the hair cells. We see these stereocilia. And depending on how your sound hits that and what direction and how fast it goes across that, it's going to change your pattern of nerve signals, right? And so tip links that connect adjacent stereocilia are going to be crucial for your signal detection in your hearing. And this is the tip link, by the way, right there. You can just kind of barely see it here because it's so much smaller and less dark. Yeah. The tip links are going to be coupled to ion channels in your stereocilia membranes. These are going to be gated by mechanical stress. And so we're literally having a impact causes signal situation. Okay? That's exactly what's happening. And that's exactly why you can actually hear if you tap on your ears, right? It's not a lot of sound tapping on your ears, is it? But it sounds really loud. Yeah. Okay, and so this is actually the mechanical situation that you're looking at. The hair cell is going to signal detection. And so small displacements of your stereocilia, these are the stereocilia, of course, bundles are going to alter tension on your temp links, and the temp links are right here. And it's funny because this is actually pretty much how a microphone works too, okay? And I can talk about that if you want, but basically these are like little springs, okay? And the tip links will act like that, little springs, the more tension on them, less tension on them, right? And this is going to work because you have these stereocilia that can separate different amounts depending on what's hitting them, okay? And so the displacement towards the tallest part here, ion channels to open and depolarize the membrane. Displacement in the opposite direction is going to cause your ion channels to close. That's going to hyperpolarize the membrane. And so right here is actually what we're seeing. Here again is our stereocilia. Right here we have our tip link, and then these are our ion channel. See that? Okay. It's literally like a little gate. Okay, and so what we're going to have is right here, we have these opening, and this is going to be greatly favoring this state when we're not stimulated, right? The closed state, because we don't have anything knocking these apart, okay? But like all springs, when you're in a real environment where you have stuff happening, it's gonna have a little fluctuation all the time, okay? And so that's actually why people can get their hearing to the point where they end up having a constant ringing sounds and stuff, because you are. You literally are, okay? That's how your hearing works, is you have a baseline level of nerve propagation, okay? And then when you're under stimulation, i.e., everybody hear it okay? Okay. I've stimulated yours. And so that's going to shift your equilibrium towards the open state, right? And that's going to cause you to start putting off signal. Now, note that you will actually start blocking that out because you're going to lose the ability to put off signal, aren't you? Yeah. You have to reset between. And that's why if you have like a really loud concert going all the time, you'll lose your ability to hear it gradually. Okay? Because you're not getting resets enough off. Okay, there's been a steady conversion from functional OR genes into pseudogenes over the course of the primate evolution here. And so this is your comparison of your different organisms, yielding insight into your sensory system evolution. And right here we have the human. Here's the chimpanzee, gorilla, orangutan, gibbon. And you can see that there's been a basically pretty steady loss, right, of our sensory functions. And interestingly enough, humans have some of the worst sensory functions of all animals. Now, there has been some kind of neat studies recently that are correlating the sensory function, how little sensory function we have, to the ability of thinking outside of our environment. Meaning, the less brain taken up with sensory functions, the more we have to do other stuff with, right? But that's very recent, and there's no telling how accurate that is. That's really way too recent. More colors can be distinguished with 3 versus 2 cone pigments, of course. Humans have 3 cone receptors. That's why we can see these 3 major color groups very well. Anybody know why we would have a more concentrated one for green, by the way? Rather than necessarily yellow? If you were to look at it from a logic evolutionary standpoint, then yellow is kind of all around us and we don't really care about it, right? The sun? What's the sun going to do for us? Not a lot, except for being able to see in the first place, right? But when we're talking about green, that's plants, isn't it? And so we need to find plants so we can eat them, so we can avoid them, so we can sleep under them, whatever, right? Red, again, a lot of fruits and stuff. Blue, a lot of dangerous stuff, a lot of poisonous stuff usually, and a few things you can eat. So most other placental mammals actually have 2 color receptors, and that's kind of an interesting differentiation, okay? This is why a lot of the other animals can only see very limited color ranges and pellets. Earlier ancestors of all animals had 4 cone pigments, as do modern birds. And so modern birds see nothing like we do at all. Okay? They would see a completely different color palette to the world. In fact, apparently to cats, we have stripes. Did you know that? Yeah. So there's apparently a UV variant striping on our skin. And so animals that can see that UV region, they would see us with stripes. So do you have stripes? Yes. You do.